Hello, my name is Ivan, and I'm gonna present my project called Gradual Domestication of Root Traits in the Earliest Maize from Tehuacán. And I wanna start talking about the domestication of maize. We know that maize was domesticated from teosinte 9,000 years ago, and the phenomenon of domestication has been focused in above ground traits or aerial traits like the branching of the stem or features of the seed, but little attention has been paid to the root system in terms of domestication and how the root traits has been affected during this process. These with exceptions, for example, the work from Amy Burton in 2013. What Amy Burton did was a wide phenotypic characterization of the root systems of several populations of maize land races and teosintes, and the main contribution of Amy was that the root system of teosinte tend to have less seminal roots. They vary from 0 to 2, while maize tend to have from 2 to 8 no seminal roots. So this is one of the difference between the root system of maize and teosinte, but also the work of Hannah Schneider in 2021. She discovered a new phenotype called MCS, or multiseriate cortical sclerenchyma. And this phenotype is characterized by the lignification of the other cortical cells. So this phenotype is present in maize varieties, but is absent in wild varieties of maize. So these are two traits that distinguish the root system of teosinte and maize. And here I present a picture of one teosinte of my hometown in Mexico. And it's also interesting to talk about the origin of Mesoamerican agriculture. This question intrigued Richard McNish and Paul Mangeldorf in the 60s. Here they are. Here is Richard McNish, and here Paul Mangeldorf. And they make a good team together because Richard McNish was an archaeologist and Paul Mangeldorf was a botanist. So they were trying to understand the origin of Mesoamerican agriculture, and they have an hypothesis in which they thought that the origin of Mesoamerican agriculture was in some place in the center of Mexico, maybe at the south of Mexico City, but at the north of Chiapas, in a place close to the center of Mexico. And they, in order to start the excavation, they consider environmental characteristics like the precipitation, because if they wanted to found all the specimens of domesticated plants, they need to look for dry places, because in humid places this plant remains degrade much faster. So they considered this, and they found the Tehuacán Valley, that is a dry place with several rock shelters that were inhabited by the first farmers in America. So in this expedition was really productive for them because they found more than 24,000 of specimens of corn. They found some cobs, some stalks, and a few root systems. And these specimens are in the collection of ancient maize of the National Institute of Anthropology and History in Mexico, and Aurora Montúfar is the one who take care of this collection. And curiously, in the book, Richard McNish reported that they found a root system of maize with seminal root system. So it's interesting because if they were aware that their seminal root number was the main trait that distinguished teosinte and maize, maybe they will develop more research in this. But in the 60s, the origin of maize was not very well understood. They don't even know that teosinte was the ancestor. So, in 2016, we developed a new expedition to the Tehuacán caves, and this was thanks to Ángel García Cook, this person here with the hat. So Ángel García Cook was a young archaeologist in the 60s, and he was in the expeditions with Richard McNish. So he knew very well the ubication of these caves. So he was our guide in these expeditions. So we discovered new specimens of corns. Some of them was a root system, and some other were cups. And Miguel Vallebueno, who is in this picture, he extract ancient DNA from these samples, and he was able to construct a phylogeny considering several teosintes in green and maizeland races in red. And what he found was that the specimens SM3 and SM10, they were in the middle of the phylogeny. They didn't belong to the teosinte population, but neither to the maizeland race population. And he looked a little bit deeply in the domestication genes like TGA1 and TB1, and he looked at the genetic diversity of these genes. And what he found was that one of these genes present the diversity of maize, but the other gene presented the diversity of teosinte. And the conclusion of this work was that these specimens were not fully domesticated, but they didn't look at the root phenotype. And if we know that the root phenotype of teosinte is different than the root phenotype of maize, so we ask ourselves what was the root phenotype 5,000 years ago. And we start looking at the anatomy of these roots. We use laser ablation tomography to explore this possibility. And to be honest, at the beginning, I was thinking that we will find an empty structure here, but fortunately, we found completely the opposite. We found very detailed structures of the anatomy of these samples. For example, if we look at this panel, we can see a very clear distinction between the stele and the cortex. In the stele, we can observe structures like the phloem, xylem vessels. We can even distinguish between early metaxylem vessels and late metaxylem vessels. We can very clearly distinguish the layer of pericycle and the layer of endodermis, several layers of cortical cells, and a phenotype that resembles to MCS. So if we have MCS in these specimens, this will mean that the domestication traits of the roots were present 5,000 years ago. So this talk about that the domestication of the roots happened early during maize. But in order to test that, we developed two techniques. We developed an staining with hydroxychloride, and in order to look at the structures of lignin here. So the MCS phenotype is characterized by increase in lignin. So we can see in red color that the other cortical cells have an enrichment of lignin. So this is a evidence that we have the MCS phenotype here. And also the increase of thickness of the cortical cell walls gives us an evidence that we are observing MCS. We developed this with electron microscopy. And yes, we have a domestication trait already present 5,000 years ago. But what about the seminal roots? So looking in our sample in SM3, we observed that the scutellar node, which is the structure from which the seminal roots develop, it was not present here because it's a very fragile structure. But we knew that Aurora Montúfar in the National Institute of Anthropology and History of Mexico, he was like taking care of this collection, and the reports of Richard McNish from the 60s say that they have this seminal root system. So we talked with Aurora, and she was very positive with us. She allowed us to see this collection, and we found this root that have the scutellar node intact, well, almost intact because it was broken in this region. But in general, we can observe very clear the architecture of the root. For example, we can see the first node, the second node, from the third to the sixth node, and we can observe very detailed the scutellar node structure. So, for example, this part belongs to the mesocotyl. Here we have the scutellar node and the primary root. And at first sight, we didn't observe any structure that resembles to seminal roots. But in order to be 100% secure about this, we did a 3D reconstruction using laser ablation tomography. And if we observe any connection between the vasculature and the cortical tissues, we can tell that we have a seminal root developing from here. So this is the scutellar node, and as you can see, there is any development of structure between the vasculature and the cortex. So the answer was no, we don't have any seminal root. We just observe the development of a root in this part, and this belongs to the primary root, and this will be a lateral root of the primary. So with the exception of this lateral root, we didn't observe any other structure, and this will mean that the SM11 specimen belongs to the distribution of teosinte, and this means that we don't have any seminal root. So some of these domestication traits were present 5,000 years ago, like MCS, but the number of seminal roots was not present yet. So maybe this increase in seminal roots was fixed a little bit late in domestication of maize. And just to explain a little bit the possibilities of having this absence of seminal root, we were thinking about this could be a result of phenotypic plasticity. And for example, Su in 2006 developed a work in which he analyzed populations resulted from a cross of B73 and MO17. And he tests these populations under low phosphorus and high phosphorus conditions. So what he observed was that under no limiting phosphatability, the range of number of seminal roots was between 1 and 6. But when he applied phosphorus stress, the range increased, and there are some lines of maize that develop 0 seminal roots or didn't develop seminal roots, and they develop until 8. The distribution of number of seminal roots increased, but the number of lines that develop 0 seminal roots was less than 5, and the probability to develop 0 seminal root was less than 2%. So we can tell that the possibility to have 0 seminal roots due to a lack of nutrients in the soil was relatively low. And we also look at some at 2 genes specifically using the ancient DNA from SM10, the one reported for Valle Bueno in 2016. And this specimen is contemporary of SM3 and SM11. And we look at 2 genes specifically, at RUM1 and RTCS. So when RUM1 is mutated, we have a lack of seminal roots, and when RTCS is mutated, we have a lack of ground roots and seminal roots. So what we observed was, in the case of RTCS, we observed an indel of 4 nucleotides in the ancient sequences in the upstream region close to the promoter of the gene. So this could be altering the expression of this gene, and maybe this could be the reason why we don't have any seminal roots. But this is just an open possibility, because the dip at which we sequence is relatively low. We just obtained, for example, 3 sequences for this region. And for RUM1, for example, we obtained that we also have an indel of 11 nucleotides in the downstream region that could also be affecting the expression of the gene. And with this, I conclude. The main conclusions of this work were some of the maize root phenotypes present in actual maize were not present 5,000 years ago, but others were present. And this tells us about that selection affected the root traits late. Well, some of these traits were affected late in domestication, and maybe the mutations in RUM1 gene and RTCS gene are the responsible for this. It's just an open possibility. And with this, I want to conclude. Thank you a lot to the lab in Mexico and United States, and I will be really happy to take questions. Thank you.